everyone, my name is Xiling. I'm a 5th year graduate student in a neurobiology lab at University of Southern California. I'm very glad to have this opportunity to talk about my research and myself to everyone. So about my research, in our everyday life, everything you think and do relies on the communication between neurons in our brain. So where this communication happens actually happens at the synapse, which is the junction between two neurons, and we call it a synapse. And the neuron 1 can talk to neuron 2 by release chemical components, which is signal molecule that we call neurotransmitter. And those neurotransmitter binds to the receptor proteins on neuron 2 to transmit the signal. And when this communication is actually interrupted, this synapse can adapt itself to maintain a stable dialogue between neuron 1 and neuron 2. So how can it do that? It has two ways, basically. So it can change the receptor levels on neuron 2, or it can change the amount of neurotransmitter release from neuron 1. And how this modulation happens, and one big question is that the specificity of this modulation is in what scale. So because in our brain, there's much more synapse compared to neuron numbers in terms of numbers. Because one neuron can talk to many other different neurons at the same time. So if this neuron can modulate the neurotransmitter release at some of its synapse while leave the other synapse unaffected. So that's the question that I want to answer in my project. So in my study, I choose a very special synapse which formed by a motor neuron and a muscle fiber. So I choose a particular NMJ. So the muscle fiber and neuron form the junction, which we call neuromuscular junction, NMJ. So I choose a special NMJ in the Drosophila larva, which one single motor neuron innervates both muscle fiber, and they are adjacent, which is amazing. It's a great model to study to answer my question. And we can do some interaction of the communication between neurons, between the motor neuron and the muscle. So we lock down the receptors on this muscle to reduce the receptor level on this muscle. So this weakens the communication, right? So that the muscle feel the weakening of the communication, and it sends a retrograde signal to tell the motor neuron, hey, release more neurotransmitters. And this actually can be heard by this motor neuron. And what is amazing that this one motor neuron can modulate only this subset of synapse on this muscle, while leave the other synapse on the other adjacent muscle unaffected. So this stabilizes the communication of this muscle, and the other muscle's communication is like original. It's not changed. And this means that the modulation can happen in a remarkable degree of specificity. And knowing that, it can help us to understand how our nervous system process and stabilize those signal transduction in our brain, and both in health and after injury and disease. So I'm actually performing intracellular electrophysiology to see how those neurotransmitter release are modulated. So I insert glass pipette into this muscle fiber to perform intracellular electrophysiology recordings. So in terms of intracellular electrophysiology recordings, the glass pipette that I use is from WPI. So that's thing that I use every day almost. And also to fill this glass pipette, we use a microfill to fill this glass pipette with the solution. So that's the most important two things that I use every day.
Mm-hmm.
So I'm not the first one who started doing this in our lab, so it's actually found out by previous lab members. And it's a great thing that we continuously using for many years. And yeah, we just use the thin wall with filament for the recording, and for the stimulation, we just without filament. And both of them are come from WPI. About my ultimate research goal, there's a lot of things in science. Science never ends. But for now, my most interesting is in finding out how synapse actually can conduct such rapid and precise synaptic transmission in the brain to process the information transfer in our brain. And in particular, I'm interested in looking for the properties and molecular mechanisms for synaptic transmission and plasticity, and how they are modulated during development and disease.